Great. Welcome everyone to the November's Reclaim Community meeting. Today we've got a very exciting talk by Cholpisit Kiattisewee, better known as Ice. He prefers Ice. Um, he is an Engineering Excellence Postdoctoral Fellow at the Institute for Medical Engineering and Science at MIT. Previously, Ice did his master's and PhD in molecular engineering at the University of Washington. He worked at VizTech, which is a research institute in Thailand, and all of this before his current work at MIT. Ice's expertise lies within the area of bacterial synthetic biology, working on CRISPR gene regulatory tool development in non-model organisms during the PhD at the University of Washington, and now his research focuses on antimicrobial resistance and engineering living therapeutics, so practically living things crafted for treating human diseases. Beyond the bench, ICE engages in a number of educational and science policy initiatives. He hosted a monthly SynBio journal club in Thai language, gathering online communities to discuss the recent scientific discoveries. And he's also worked with SynBio for All Africa as a founding member and an instructor, helping the team establish free online courses, which started in 2023. ICE also engages in the bioeconomy policy discussion, especially in the deployment of engineering biology technology into the natural ecosystems. Today, he'll be talking to us about translating laboratory experience to accessible education. So that includes his experience in advancing CRISPR gene regulation research and his path to applying this and his knowledge to gain for accessible education. So without much further, I'll pass the mic over to Ice to tell us a bit more. So please thank Ice and welcome him to today's call.
Hi, everyone. It's my pleasure to be here. Let me try to set up the slide real quick. I think, let's see. I think I got the screen up now, I guess.
It's still loading for me. Okay, it's up now. Perfect. Thanks, Ice.
Yeah. Very nice to see a lot of you here. I would like to mention that the action or the meeting that made me aware or get to interact with a lot of the Reclone community members is when I go to the Spirit of Asiluma conference earlier this year in February. And I think I met Jenny, I met a lot of other people who has been actively involved, like including Sibel, who has been the co-host for the meetings and for a lot of times. And learning about what Reclone is trying to establish and trying to improve the accessibility in science. I'm very happy to hear about the community, what the community is going forward to. My background is on the microbial synthetic biology, and I'm trying to do more things in education and also like in whatever scientific communication and other fun things. So what I'm trying to show you here today is going to be like one of the most recent publication that came out from my PhD. I just started my postdoc for just 1 year, so I don't have a lot of results accumulated yet. So I'm going to share something from my PhD first. Those are one of the basic things that I feel like that project sort of taught me a lot in terms of like how to adapt whatever experience that I got from working in the lab and try to do a lot of other things. So in this case, I'm trying to like tell the story more on the trying to make these experience useful for the accessible education. So for the talk today, I think I may have some animation. All right. Yeah. So for talk today, there's going to be like just two main topics. I'm going to talk about the research experience in the bacterial CRISPR first. It's going to be more of the science talk where I'm trying to explain what is CRISPR activation, which is the key ideas of what I have been working on during the PhD, and also on our quest to achieve the synergistic CRISPR activation in microbe, which is turns out to be non-trivial, and there's a lot of failure along the way. There's a lot of learning we've got, and then I'll try to like see if I can adapt those ideology to the educational approach. And two things I want to emphasize just right here. I may not like pops up these text that that often I really appreciate, the community effort to make sure a lot of things is like open, especially for the OER, the open education resource, and also like make sure that like whatever education we're trying to do is very context aware. That was like very important when you're working in a global mindset, similar to how the clone is operating right now. So you may see, these bears, from time to time. These are actually my character design when I was working. And, when I have these, it sort of like helps me a lot on trying to tell how comes, I want to like represent myself in whatever system. So like put it into a lot of the figures I make. So this is like, a representative journey of my own journey, and I call this slide like the origin of ice. And I really like making things fun all the time. And actually, like, I was from Thailand. I was, born and raised in Thailand, stayed there for almost like, 25 something years before moving for the PhD in Seattle, working at U of Washington, working on the microbial biology. So actually, like when I start my scientific career, I started with organic chemistry, then I moved to enzyme chemistry. There was more biochemistry, and then I know a lot more about the microbiology at the time. Then I try to like, combine them and get more in depth into the microbial acidic biology later on. And right now I'm trying to reverse those training I have at MIT, trying to make the microengineering tools and also the living therapeutic applications. I'm working in Jim Collins' lab right now at MIT. So, there's a lot of great teams, members who can, talk and chat about a lot of projects. So that's one of the reasons I really like talking to different community, because that's how we can share ideas and share perspective on different things. Yeah, so just so that we can go right into the research story right away. So when we talk about the CRISPR tools development, I really like showing this kind of metaphor of comparing the central dogma, which is composed of like DNA, RNA, and protein, with this kind of like data manipulation or whatever things that we are familiar with, like, in a phone and computer, they have the data storage. We want to move things around. We probably like, transfer it to something on the cloud or on a thumb drive. And then if you want to run it, we just turn into an actual application. So those are similar to like how the DNA and RNA and protein are operating. The good thing with the CRISPR system, if which I bet like everyone here has heard about it to a certain extent, is that it can be repurposed to control a lot of the parts, especially in the DNA and also the RNA. And that can be used to control either the replication, transcription, or translation processes. And different type of Cas proteins that can be used. And in this case, in my lab, we've been focusing a lot on the Cas9, particularly on the dead Cas9, where it doesn't cut anymore, but can still retain the ability to bind to the target DNA. So with this ability to bind to any target DNA, guided or programmed by the RNA structure, which you see from these. I'm not sure if the cursor is showing, but this structure looks like a hairpin, a loop, or something like that is what we call the guide RNA, where these RNA will be complementary to the DNA target, and that's how we get into the actual target gene, either on the plasmid or on the genome, whether like it's something that you put into the system recombinantly or something that has already been there in the natural system. So with this, what we are trying to focus a lot more is to focus on the transcription system. So these are, the kind of like fun metaphor that I really like to make all the time. So assuming that, I'm on a horse and I'm trying to go through down the road, trying to go into, whatever supermarket, trying to buy something. In this process, if I run into whatever roadblock, similar to how, the CRISPR protein can sort of bind to the DNA and block whatever transcription, well, the work of these RNA polymerases try to read the whole gene. This is similar to like, trying to interrupt whatever work that you're trying to do. So another metaphor that we like to use all the time is like, it is similar to a traffic light, like you give them a stop sign, and that means things are likely gonna stop. Alternatively, my PhD lab is working a lot on the activation process, where if you, go back and compare with the interference or like, the stopping process, there's a lot more parts in it, because you have when you're trying to turn something on, or when you are trying to facilitate the process, like asking an engineer to help make you go faster, you may have to put some more things and trying to study the rules on how you can actively or effectively activate or turn things on. So those are the caveat of the bacterial CRISPR gene regulation, because if you're trying to block or get rid of something, it's kind of simple. It's a lot easier to break things rather than build things from scratch. So that's kind of the same metaphor with working around these things. So with these kind of annotation of like the CRISPR interference or CRISPR activation, which I may refer to CRISPR-I or CRISPR-A sometimes, these are the main system that we were trying to do in my PhD lab at University of Washington. So in this system, having these established, we have one key question, which is like, if there is a CRISPR activation available, and we have the ability to bind to any DNA target using this guRNA, RNA, DNA complementary sequence to program or to place this CRISPR complex into anywhere in the genome, can we achieve some kind of like improved activation by using different activators or different system to make these gene activation processes being a lot better than just just the additive effect. So the definition of the synergy is that like when you take effect of like system A and system B can combine together, if you have these summation of it, that's something that we can refer to as additive when we talk about things in biochemical system. If you have a lot more than you expected, then we refer to that as a general synergy, when two systems are working much better than we expected. And this is something that has been shown before in bacteria, when there are two transcription factors binding to the DNA and trying to activate these at the same time. So the fact that they can do this synergistic activation is because they can bind to different part of the RNA polymerase and trying to recruit them and make this recruitment a lot stronger than just having one of the protein or transcription factor do it alone. Interestingly, the synergistic CRISPR-A has been very well established in the eukaryotic system since the coming of age of CRISPR in around like 2010 to 2013. The CRISPR interference and activation has been like one of the key tools to study the mammalian and also the eukaryote gene regulation system, where you can just target anywhere in the genome. There are a lot of systems being developed right here in the CRISPR-A in eukaryote system. However, there is barely any synergistic report for the CRISPR-A in bacteria. I have to declare that there are a lot of teams, including our team, my PhD lab, is trying to achieve this synergistic CRISPR-A. But most of the report has been reporting something that doesn't give any improvement, or something that may lead to a lot of like failure along the way, where you get like reduced gene expression rather than increased gene expression. So that's why we think that if we can do it systematically, we might be able to achieve this kind of synergistic CRISPR-A, given that we know enough about the bacterial CRISPR activation system. So before moving forward, what we try to sit down and like make sure that we understand this properly is that we have to make sure that we understand and also characterize the system systematically. Why? Because when you're trying to make something work and also work fine and effectively, you have to understand how it behaves. A very quick question on how to use the system is that like if they have different activators to recruit the RNA polymerase to the promoter, then you may want to figure it out how do you recruit them and how do you put them into the proper position. So two main questions that we have here is like how do we link this effector protein to the Cas protein, to the dCas9. We can either, one simple way is to put into the Cas9 as a protein fusion right away, or the other way that our lab has been preferring a lot more is to put into the guRNA and use the RNA binding protein to interact with the RNA and take the effector protein on top of the Cas system later on. So with these number two recruited by guRNA, the pro of it is that you can program the guRNA to be having this effector or not by just program the RNA part as well. Another key question is like where they should be located, because these activators, when they work natively, they actually bind to different sites and they have like different active sites that can activate the gene expression. So these are, another key things that we have to be aware of. So the very first thing that we believe that we have to do, and we did spend a lot of time, maybe like, 70% of the time we work on this project, and these include, 3 or 4 grad students working together, is that we are trying to characterize the system in a very close system, in a very close like test set as much as possible. So we chose these 4 CRISPR activation systems that has been developed both from our lab and from other labs, and try to characterize them, trying to make sure that they have the as similar promoter, like the reporter context, that we are trying to activate the gene as much as possible. So we have this system where we use the adenosine promoter, which is one of the standard systems that people have been using in the bacterial SynBio a lot. And we have this other group of promoter where this spatial PSPF system can be used to activate the system. So the reason there are 2 systems is because like this PSPF system is exclusively acting on this group of promoter. I'm not trying to go too far in details, but you see that there are some range of the sites that CRISPR activation system can activate. And there are different efficacy and also different degree that they can increase the gene expression. So with that in mind, we just try to think about, with these different CRISPR system, what is the different ways that we can use to combine these 4 different CRISPR systems together. So I'm gonna go quickly one by one on like the ideas that we are trying to use. So the note that I put up here is actually something that the eukaryotic system has been using so far in trying to achieve the synergistic activation. So what we do first is a tandem protein fusion. So if we have this RNA recruitment system and we put one activated protein on the left side and the other one on the right-hand side, then we might be able to recruit 2 activators at the same time. However, what we see is that if you look at the green bar and the orange bar here, which is the activation level of the activator 1 and activator 2, what we see when we recruit both of them is these purple bars. And they just go completely from maybe like 20 to 30-fold of activation to less than 2-fold. So we actually abolish the ability to activate instead, rather than just like improve the expression. We thought that there might be something about the protein folding that may interfere with the functionality. So we test another system where we put these recruitment, or like multiple recruitment, using the protein-protein interaction, in which like the folding of the protein may not interfere as much. And we also don't see any improvement. So we were pretty perplexed, when we tried this system. But it could be that, it's just one of the way to combine these CRISPR activation systems. So maybe we can try a couple more. So the other approach that we try is that, like, let's say if we have the CRISPR-Cas system, and I mentioned a little bit earlier that when you're trying to put the activated protein onto these CRISPR systems, you can either put it by fusing to the protein, like the Cas9, or recruit them by the guiana. So in this case, we try by recruit 2 different systems: one by fusing to the Cas protein, and 2 by recruitment by the guiana. So these are the system that we found that it worked best when they fuse to the Cas9 or fused by the guiana, and we just try to use the base of its kind. What we found is that, like, when we recruit the 2 system and we also express the 2 activation system, we actually see some decrease in the activation. So this is a sign that when we're trying to achieve the synergistic effect, we actually see some interference or some antagonistic effect, which is opposite of what we're trying to do. So there's something very complicated going on in the bacterial system that we don't really understand on how to effectively increase the transcription activation using the multiple promoters. I want to note that there are other systems, like recruiting 2 different things. We also like put that into the paper as well when we're trying to work around it. And so far, none of these approaches that we have tried in the span of maybe 2 or 3 years really work great. However, when we are about to stop this campaign, we get back to reconsider that, like, actually we have this activated system, the PSPF, this pink thing here, that was doing pretty okay in a different set of promoters. And if you're trying to go for the different activation, we may want something that have very different mechanism or different target site to recruit or interact with. So we just go to our base CRISPR activation system, the success, which is green right here, and the pink one to try to combine. And so you have to know that, like, this success doesn't work on the type of promoter that we are trying to use here by itself. So we actually, try to put them to work together, and that might be something that we can work around that as well. Oh, I'm sorry, I forgot to mention this thing. We thought that, like, if we recruit two things and they don't really work, we can also split them into two different Cas proteins. We tried that, also doesn't work. So, like most of what we've seen so far is actually antagonistic effect instead of the synergistic effect that we're trying to look for. So that's when we come to the PSPF system. So this is probably like the last science slide I want to show. So with this PSPF and success, which is the base system and the most different system to success, is when we test this kind of system out by keep one of the activation, which is also at a different site, different preferred site to that of the success system. If we keep these around to keep activating the basal activation and just thread these second activation system around the different location, what we found is that this is the base system where we have almost 2x improvement in terms of activation. So it may sound like not a big improvement, but if we calculate the actual improvement from the basal level, is actually changing from 10-fold to 20-fold. So that was a significant change in terms of the fold change that we're trying to use in the further synthetic biology application. So this is something that we found kind of interesting, that we get into this kind of system. However, the two-fold system is only applicable to this type of promoter. And while we're trying to expand a lot more, we found that there are some other technical limitation. For example, this type of promoter is available only in less than 3% of the bacterial promoters in E. coli genome, and there's a lot of like other bacteria where these type of promoters are not really characterized and doesn't have a lot of good bioinformatic systems to evaluate which type of promoter and what is the defined rules that we can use further. So we kind of like just wrap it up here and try to see what kind of story we can come up with, and we just share that later last year. So, in this case, it sort of work, but we are trying to be extremely careful when we communicate this out, that it works, but with some caveats, it only act with these small group of promoters, and there might be some interaction that came from just the Cas9 binding alone that could be affecting this whole system as well. So, and you have to bind exactly at the right position, otherwise the effect is very minimal, like 1.1. 10% improvement is very difficult to solicit it in the synthetic biology, bacterial synthetic biology system. So what we learned, the two big things from what we have here is that first, it seems like the least likely combination that we've been working on is the one that seems to work. We don't know why, but it seems like, this finding will never be possible if we don't spend time on trying to characterize the system properly and systematically. So these are the two key findings that we want to deliver and also communicate to the field, that if you're trying to do these kind of processes, you may have to consider the systematic approach, and also whatever combinations that might be possible as well. This one is the least possible combination that we would like to try, but it seems like it's the one that give us some ideas that this is the process that might be possible to get the synergistic CRISPR activation in the microbe. So, throughout the whole project, the high-level takeaways that we can share is that most of the times, if you're trying to do any processes in science, the expected results may not be the actual finding. Sorry. The expected results may not be the actual findings. Like, we believe that like this kind of synergistic effect theoretically should give us, some kind of improvement. But a lot of them actually give us the opposite, where it show the antagonicity, where they just like interfere and make all the functionality just like decrease rather than increase. And the other note that we want to share is that we perform the research or whatever experiment systematically there's more likelihood that we're gonna learn something because there's a lot more controls and the positive and negative that we can combine and try to learn what we've got. And the final thing that I felt like this is one of the work that I'm very proud of during my PhD is that you see not a lot of these work that I'm sharing has extremely good positive data, right? But if we decide that like, well, we don't have a lot of great data, we didn't make a big change in numbers, maybe we don't really have to share it, then no one really knows. And there'll be someone trying to go to maybe a similar path to us, trying to see if the system work or not. So we believe that like, since we do it very systematically and we have a good learning that we have come out from this kind of work, we should be sharing them appropriately with the caveat and with the remarks that like, hey, it's hard. We try a bunch and these are not working. If anyone should be try, if anyone were to try these systems again, they should try to work around the system that's not exactly like what we were doing. Otherwise, they may fall into the same kind of trap that we have found, even though we're trying to be very systematic. So another note I want to give here is that like this is the work that was led by maybe 2 or 3 different generations of the grad students that was in the lab, and it's sort of like tell a lot of the perseverance and work that if you were to do things in science, it may take time to build things, it may take time to work around, and it may take a lot of conversation to build up these different perspective on how you should be doing things. And the possibility of doing science is like very broad, and you only have like the finite amount of time to work on some kind of problems. So I think these are some kind of learnings that tell us not just about the scientific findings, but also on how do we work around the science to actually get something that was meaningful and that we just teach us and also teach the community on what we should be doing things as well. So, with this kind of science that I'm sharing, I'm trying to see what is the proper way that we can take these kind of takeaways and learn it and apply it to the educational approach, especially in the STEM education, because like it's so difficult to teach things, particularly in the biotechnology where if you trying to learn how to set up the lab, trying to do the PCR reactions, and you don't really have the system, how are we going to do it? So what we want to, or I see the system that I really want to emphasize here is that we were talking about education. It's not about just taking lecture. If you go to the biotechnology class or biochemistry class, and the only thing that you're doing is taking lecture and also trying to understand things, then you learn, but you may not be able to do the actual things that will be required to execute when you go to the professional workflow, something like that, right? So there's a lot of system, and it's not even just the lab, right? I mean, what we've learned, or what my friend and I in the lab learned, is by trial and error, by trying to design something that will teach us and also teach others on how to work on things. So this is, one thing that I want to share in terms of like when we translate the research experience or research training into the education, is that education doesn't mean just taking lecture. We have to take different forms, different format, a lot of discussion to work around. The other things is that, if you want to, what we learned that we have to do things systematically in order to learn things. If we want to make sure that the education and platform is going well, we have to make sure that we build this platform kind of systematically as well. And by systematically, I mean that, if we were to share things around different places, let's say I'm from Thailand and was working with African people, at some point I'm trying to make this thing applicable globally, then I have to make sure that the system is set up properly. Um, so there's a lot of discussion in my lab when I was trained early in my career. One of the remark that was told, and I still remember to this day, is that like you spend time on the desk trying to think very hard on what you are going to do and why you should do it, then you probably save a lot of time in the lab, because you can get rid of a lot of something that may not tell you or may not be that necessary to do in order to get you to the actual answer. So this is like, something that was equivalent, because if you make the system or the platform systematically applicable, then it may not matter that much if you were to apply it to different system, as long as you're aware of the context. That might be a small difference there. And finally, we learned that if we don't share things on our finding, someone may get lost again. And this is, an emphasis that if we were to do anything, if we were to do any educational resource, any course, we want to make sure that everything remains open and accessible to everyone. So, I showed these three figures that I was working with in three different communities in Thailand, where I came from, and where I can speak another language. In Africa, where we have a pretty good team that I collaborate with, and also at the global level. So these are some notes that I've been working on. So I did work a lot with the Thai community, and I did, in a different format, like the podcast, we talk about, how people come to do science. Is it okay? We have to stop doing science early and move to a different stage of life. So that's kind of a conversation that people don't hear that often from the scientists. We host a monthly journal club where we come in and try to do the journal club similar to how scientists do. And we try to like make it open so that people see that like when we do journal club, it's not that we try to read the paper and understand everything. We want to share what kind of struggle we have when we go through the system. And at the same time, we also like work on the blog post. It's not just me writing this thing, but we collaborate with the Synthetic Biology Consortium in Thailand to work on some kind of like recent research roundup that can help people trying to interact and trying to bring people in to learn about something new that might happen in the near future. So these are something I was working in Thailand, and I was, training myself and also my team on how to make sure that we can have access across the different media forms. So you see, there are some that was based on voice alone, there's some that based on the discussion, there's something that was based on just writing. So these are something that teach a lot of people in the team on how to work around this thing. And technology is another challenges. The good thing in these days is that there is a lot of open source learning materials that help me teach me how to draw proper figures, how to edit the video into the proper length, something like that. So with those kind of like training that I had, I was able to help the community called SynBio for All Africa a lot more. So with that, we sort of like started aiming to work on the first in-person work in Africa. So we have some kind of like um, advice or work something that we work together with a international volunteer to set up some kind of like research goal, and we will have the people who can work on the actual wet lab project in Africa. And right now we just established some initial plan in Uganda so that we get to work around the plant genome sequencing. And what we're trying to do is we're trying to see if there is something that was overlooked in the system. So in this course, we got support from the Experiment Foundation, so that we have some budget to do some genome sequencing. But as a volunteer like me myself, I wouldn't know what is the most important thing to sequence or to figure out in Africa. So we spend the whole course trying to teach the people who take this class what is genome sequencing and what genome sequencing can tell us, especially in the synthetic biology, and what can it expand into to give us more values added when we learn about these. And at the end, we have the community decide together which one should be the one that we go for. So we got, the amygdalina, sorry, I lost the species name on my lip right now, but it's a plant called bitter leaf that what I was told by the people in the East Africa is that it's something that if you feel sick, people usually go and take the leaves and just mince them and use that as some kind of like, herbal medicine. And not a lot of people outside of Africa are aware about this kind of medicinal plant that people have been working on. So if we know more about, what is the composition genetically, that means like there's a lot more potential for the African community to learn more and expand on this research. So that's the reason that we go for this thing. And another note that I want to, acknowledge is that we work a lot with the Engineering Biology Research Consortium people in the US. So EBRC is an organization, a consortium that have a lot of US SynBio engineering biology people to come together and work on a lot of policy, on the science, on a roadmap. And we have the student postdoc community as well, where we come in and volunteer to work around this course together. So finally, I was also, taking the course that was called How to Grow Almost Anything. So sometimes we call it HTGAA, where we work around trying to teach, like having the great scientists coming to teach people in more design way, where the non-scientists can take this course together and trying to learn how to work around the different system. So what it just work with the team is that I think we need to find a better coloring process so that we can do an easier and more colorful design. So what we did is that we curate a lot of different fluorescent proteins or colored proteins and try to put them into the same systematically designed, plasmid, bacterial plasmid system. And these are the different colors that we tried. Some of them are very brightly fluorescent. Some of them give a different color palette. On the left-hand side came from the UV without filter. On the right-hand side is with the filter. So the color will look a little bit different because it cut out the blue light. And what we see as a potential is that, you can make this kind of like beautiful figure if you have enough colors and enough shading. So, that's some kind of potential that I want to share as like the education approach. Like if you know how to systematically make things and make a lot of, like collection of things, like in this kind of fluorescent proteins or colored protein setup, you can actually achieve this kind of beautiful design, which, not a lot of scientists is trying to think genetically how to change this, the genetic cassette so that you can have these different shades of green and red, something like that. So, I want to wrap up and share and acknowledge everyone that, I work with a lot of people, with the team in Thailand, with the team in Africa, and the team here in Boston that was trying to aim for, a global engagement in terms of education and trying to make the education accessible for everyone. So with that, if there is any question, if there is a discussion that we want to talk about, I would be happy to, share with everyone here. And thanks again for your attention. If there is anything that I can help with, please feel free to reach out to me. I would love to interact more.
Thank you so much, Ice. I can see already there's questions. Eric, would you want to go ahead?
Sure. So I saw in one of your slides you show where it was kind of partly in Thai and partly in English, but I guess one of my question, one of the things that we have at Addgene as far as resources, we have our CRISPR ebook that is, we have the English version, and then I think we have a Spanish and maybe a Chinese version. But the challenge with translating these types of things and to make them more accessible to people is that actual translators are expensive. So I'm wondering, like, have you had any experience with like using translation software or anything like that to do this, or did you do this on your own, putting these things into Thai? And I'm just wondering, just at a high level, we'd love to make these our educational resources more accessible to the non-English speaking world. So I'm just wondering if you or anybody else here have thoughts about the best way to go about doing that that's not gonna be too expensive, basically.
Yeah, absolutely. I think that's a great approach and ideas, Eric. So I can share one thing about these, content in Thai language is that it's extremely challenging for some languages to work around science. So there's a book that was dedicating on like, why is English is kind of like the language that was used for this kind of scientific system, because like it has, some simple approaches or context that we can use. Like for example, what I can share is that in Thai language we have 44 syllables, we have 44 characters, we have 30 vowels in terms of writing. So it takes a lot more time to translate and to make it readable to everyone. If you put it into, let's say, some large language model, whatever software that can help this translation, it's going to turn into something that can be read, but not that accessible. Because if you work in the actual topic, you will know what kind of translation or whatever English, word that it was translated from. But if you have no clue, and you need something to be more accessible in terms of like being simple to someone who are like more beginner to the topic, then you will need people to work in. So getting back to the software, I think right now, everywhere around the world, we've been using a lot of the automated translation. And for the translation that is very friendly to, the language family, let's say we translate from English to French, English to Spanish, it's going to be a lot easier than translating English to Chinese or to Thai. And what we found is that, like, even though it's not perfect, it's a lot better to use those tools as a starting point. And then we can have some volunteers or some people to come in and see, like, hey, if there's something that is extremely bad, or like, it's going to be misleading a lot, then we should have people come in and modify it. So that's how we work around this process. And that's why we try to make this journal club being in Thai. Because when we share about these things, with the other students and postdoc and in my friend circle, we believe that, like, talking in Thai show the struggle, and we try our best to talk completely in Thai, not using half English, half Thai, or something like that. So that's another interesting challenge, and also something that we would like to work more around that. So last thing I want to share about this is that, if there's anything I can help to bring on people on a different native speakers on different languages, then I would love to help connect as well. I know that I know a lot of community in Thai. I have a lot more friends in the Asian countries and continent where they speak different languages as well. And I think they would be very happy to see more Asian contents in these different languages, so that it's more accessible to them.
Great, thank you.
I'm sorry for the long answer.
No, that's really helpful.
It's definitely something that we have considered as well, because I mean, we are very fortunate to have Sibeli on the team, and she's been working a lot with the LATAM community over the last year or so. So she's posted blogs in Spanish and Portuguese for us, as well as English. And you do need that kind of person who does know the language well to be able to kind of fine tune what we present, even if we go for any AI, if we go for any kind of AI software to try and do the first pass, let's say. And yeah, I think we also had one of our other community members, Joseph. He also has a group out in Syria and the Middle East, I think, where he was helping also with translation of protocols and resources in that way. So it's always really helpful to have these non-English formats. I suppose a follow-up on that is, how do you find time to be able to run a podcast, a journal club, blogs, wrangle the volunteers to help alongside your day job?
Yeah, that's gonna be a very bad answer. I didn't really sleep a lot. And that's maybe like the only, reason I can still keep doing. Another thing personally that I was able to do, these different skills and work around, is that, like, I keep doing it as I grow up. It's not that, like, hey, I want to do a podcast, I'm going to do it today when I'm a postdoc. It's going to be impossible to learn how to do video editing or voice editing ever when you have to work, like, 8 hours or 12 hours sometimes in the lab, right? So it's partly because, like, I have some interest on doing these things to begin with. Like, I did learn how to use like the drawing software to make these kind of, like, figures. So these are what I draw by themselves, and it feel like if you have these skills to work around this kind of art approach, it's a lot easier to adapt and make them better. And there's also, like, another another caveat on how scientists work. So when we work on the paper writing, when we work on anything that we want to put it out, we have a lot of peer review process. There was internal peer review. We have, like, the other kind of peer review. I know that, like, those are very important for something that's going to be, on something for publishing. But if you want to do journal club, I asked everyone in my team, like, hey, can you try to spend maybe just 2 to 4 hours on this paper and then share it. And if you don't know, say you don't know. So this helped a lot to sort of like frame that, hey, we're going to talk about this paper and we're not going to overspend the time or effort to work around these. And if we don't understand, then we'll be honest that we don't understand within the given time that we have, because everyone has limited time, right? Asking the friends to spend like 2 to 4 hours on a paper that they are interested, but not the day job, is a little bit of an ask. And I think these are the key things that I found. One is to make sure to like connect with people who have interest and have some training prior to coming to do this thing, and also like set up the clear dedicated time to work on these projects, so that we make sure we don't get tangled with a lot of like excessive work and affect the day job, and also affect the quality of the work that we are trying to put it out. So setting the bar is something very important. And I feel like scientists are not that trained to accept something that is not completely perfect, especially in the writing format. But I think these are some skills that a lot of people in industry, they were forced to work with the deadline a lot more than us. Yeah.
To an extent, 80% is basically good enough, and then we can
Absolutely.
refine.
Yeah.
I mean, obviously we've advertised about your talk on the forum, but it would be great if you could share any resources, for other people who are interested in running a journal club, video editing. It'd be great if you could share those resources there if you have a bit of time. Um,
Yeah. I can post on the on the community to start a thread around this discussion, so that you have the proper links. That just use common tools. I use YouTube for the web streaming. I use the open source to use my computer to share the screen, similar to Zoom. I sort of like use the Zoom software to record, and just in case there is any post editing. YouTube also has some kind of like simple trimming process that you don't have to learn a lot on how to work on things, and that's very easy to start. And then if you want to go into the professional setup, then there's the set of like things that you may have to learn first. But after you spend that kind of time to learn, it's more like when you try to learn how to do bacterial transformation for the first time, it takes a lot of time to make it work. But once it works, you sort of know how to do transformation, and you'll be fine. The next time it's going to take half an hour to do this, something like that.
Definitely. It's good that we can curate these kind of resources for the wider community. I'm just going to throw it out there to folks who are on call. If you have any other feedback, comments, questions for ICE, we've still got another 5 minutes or so left of the meeting. I mean, I think if folks are thinking, one of the things you mentioned earlier, Ice, about having a sort of well-defined scope and just checking people, do you have time for doing things, is always very, very useful. Like one of the things that Sibeli and I have talked about was having, for example, a community meeting, but specifically in Spanish, which does preclude me, I recognize, but it'll be useful because if, as you say, having kind of a half English, half Thai, or half English, half Spanish can be very tricky. So I'm kind of grateful how patient people are to join all these fully English community meetings. But definitely, if we have more support, I think this will be able to spread more the kind of the open science, the stuff that we're working on here, more broadly to someone's native tongue, really.
Yeah, maybe like a small thing I want to add to that is like when I work with the How to Grow Almost Anything community, we realized that the time zone is another thing that is very important when you're trying to set the meetings. So the Asian community, we found that like is going to be a lot easier if we can find some people who can represent them and like make sure that things are uploaded in time, so they don't have to stay awake at 2 a.m. if they have to listen to the class like live. So that was like a big change to people who are trying their best to attend and still keep their day job operational. And I think like having the sub community is sometimes like extremely useful. Sometimes it's too far if you don't have too many people, and it turns out to be like just a few people running things. But if you have enough members to set up like a subgroups, I think that's something very useful. That's also like something we learn from research, right? If you have a big group and we work on a similar projects, we probably want to connect and stay together so that like we get very like related response and to the point on the topic.
Definitely, and I can see already that Eric has posted this detail, so feel free to do connect and see how you can collaborate on this. And on that note, I might do a quick call out because I have been prompted about this at one point previously. We do always look for volunteers to help with the Recon community, and in the background here, we're putting along a list of tasks and like contributions from like small bits to large bits that folks can take part with. So do look out for this in the next month or two. If you guys have a bit of spare time that you would like to help contribute and kind of grow the resources, the educational tools, and everything we have in the community. Equally, feel free to also suggest things that you think that we're not doing or not doing so well that would be more of a benefit to the community. I'm going to do a last call out. Does anyone else have any last thoughts for ICE? If not, I would just like to thank you again ICE for speaking with us and you all for joining us today. And we look forward to seeing you at our December community meeting.
Absolutely. My honor to be here, and we really appreciate all the work the community has been working on. I think like the more we are connected, the further we can go. So
Perfect.
and
Great.
thanks
Well,
everyone again for staying, and I hope we can connect more.
definitely. We'll keep in touch.
Thank
Thank you again,
you.
everyone. Take care. Bye.
Once again, take care.
Thank you. Bye.